Welcome to the Proteomics course. In today's lecture, we will talk about two-dimensional electrophoresis, image processing and data analysis. So, as in the last few lectures, we have discussed about two-dimensional gel electrophoresis and in fact, various type of gel-based proteomic techniques such as SDS page, blue native page. We have talked about different type of new advancement like off gel fractionation as well as two-dimensional gel electrophoresis. We also discussed in very much detail about two-dimensional electrophoresis workflow. How can you perform the two-dimensional electrophoresis based experiments step by step, various details, nitty-gritty of performing protein extraction, different type of challenges associated with the sample processing, followed by how to perform isoelectric focusing and then how to do SDS page analysis. In between these two, what is required to make these strips compatible for doing SDS page, which was discussed in the equilibration steps, and then the second dimension separation using SDS page. Now, once you have run all of this, performed all these steps and run the gel, after that you want to visualize your spots. So, you need various type of staining techniques. So, we discussed different type of staining methods which are available, and then once you stain those gels, you can visualize those spots. You can see how many protein spots are visible on the gel, and then those spots can be further scanned for image processing. Now, a good scanning, image processing and followed by a data analysis to obtain some meaningful information from these gels is very challenging. So, in today's lecture, continuing our focus and theme on two-dimensional electrophoresis, we will discuss about image processing and data analysis, various type of challenges associated with the image processing and what are the steps required to perform data analysis. Now, when you have run a very good 2D gel, you will expect an image, a representative gel shown here, where you have well resolved spot throughout the gel. This is a gel image shown from serum protein extracted from human and those proteins separated on the two-dimensional gel. Now, if everything goes well, you can see the well resolved spot throughout the gel and the whole proteome can be well separated. But many times this is not the case. Many things go wrong. Many times the salt will have some interference, nucleic acid contamination will have some effect, and you can see various type of artifacts. You can see different type of vertical as well as horizontal type of streaking on the gels. These small details make big difference when you are doing the gel analysis for the image processing part. So, how to analyze two gels by using software is not so tedious, but when you are talking about many gels where you have large number of controls, large number of treatment, then it becomes very challenging, because all your gels should have run at the same time with the same different migration. But many times that is not the case. You cannot run 50 gels or 100 gels in a given day. So, there will be some manual artifacts, some day-to-day variations. Now, on one hand, it is good idea to keep all your parameters very consistent, so that the gel migration, all the protein resolution will be of the same parameter. At the same time, even if your all the gels have not run exactly same length, still the softwares can help you for the image processing. You need to do the various type of image processing before you analyze your gels. Now, we will talk about different type of image processing tools, how to first prepare the images for data analysis, and then how some of the commercially available softwares can be used for obtaining very meaningful information from these gels, whether you can see some sort of differential regulation of the proteins, up regulation or down regulation, or you may have some unique proteins emerging on your sample type, your treatments. So, these type of analysis will be performed on the software, and then followed by one need to do the statistical analysis to obtain confidence in the data, how meaningful this information is, and whether I can proceed with this spot for further protein identification using mass spectrometry. So, all of this details require very much practice. One need to really spend hours on data analysis. In fact, running gel sometimes is much easier as compared to doing very meaningful data analysis. So, to discuss about details for various challenges which are associated with analysis of 2D gels, the image processing tools, as well as what are the details required for image processing by using various commercial software. Today, I have invited a guest here to discuss the image processing and data analysis by using commercial software. So, today we will have Dr. Srinivas from GE Healthcare, who will discuss the how to analyze gel by using image platinum software. So, during this course of discussion, we will talk about a stepwise procedure, how to take this scanned images, the raw TIFF file, and then align those, make the match sets, and then followed by do the spot detection and various type of manual refinement for image processing. And at the end, we will discuss about how to obtain meaningful information by using various statistical analysis. So, I will have a discussion with Dr. Srinivas now. So, today we have Dr. Srinivas, an application specialist from GE Healthcare, who will be discussing with us the challenges of image processing and how to perform the two-dimensional gel electrophoresis, image processing and data analysis. So, welcome Dr. Srinivas, and we would like to initiate discussion with you on image processing.
Hello Dr. Srivastava.
Hi.
Today I would like to explain about the workflow of Image Master Platinum. Now when you can see, it is very important software to analyze 2D gels, especially when we have the multiple number of gels. It's very impossible to analyze manually. So this software plays very key role in the analysis part. So as you can see, there are the majorly two modules are available. The exclusive Image Master is a flexible solution for the comprehensive visualization, exploration and analysis of 2D gel data. Two modules are available in the software. The first one is Image Master 2D Platinum, and within this we have another module, Image Master Platinum, which is enabled for the DICE application also.
So, it means like the same software can be used for the traditional or regular two-dimensional gels, as well as it can also be used for the advanced techniques such as difference in electrophoresis.
Yes, it can be used, but up to some extent you can able to use. If you want full-fledged DICE expression, DICE analysis, one should have the decider. This is what for conventional 2D electrophoresis, Image Master Platinum is the better tool to analyze the gels. So if you can see the overview of this particular IMP software, there are three major parts. One is the complete icons, where you can see the complete the file tools as well as edit, view, and so these are all the views. And after that you can see image view. These are all the images at a time which you are visualizing, almost all 12 gels at a time which we are visualizing at particular place. And the third view, which we can see all the number of gels which is present here, the total number of gels which you can see here. These are all the major visualization part in the software.
So this is, you are showing the interface for the software Image Master Platinum.
Yes, this
where
is
three parts are shown in the PPT.
Yeah.
About various type of tools which one can use.
Yeah.
How the images look like, and then what type of parameters software is going to display.
Exactly. Those are the three major interfaces here. And you can see even workflow in the left hand side. Once we go through the software analysis demo, there we can see the complete details of for this particular feature also.
So software is made in such a way that it can walk through the stepwise
Yes,
automatically.
yes. Then if we can see the complete workflow of this particular software, first we need to add images, then edit gel images. The editing is very important because when one during scanning one may have some extra portion of the gels as well as the some streaking of the gels at the cathodic end or anodic end. These are all should be removed from the gel.
Or even
So
things can come from the staining, right?
staining even,
artifact,
yeah,
or
artifacts,
even
yeah.
some dust particle on the scanner, all of those small.
Yeah, all these
Even
should
many
be,
times, even water sometimes.
yeah, it gives
Traps
yeah,
inside the
exactly
gel, right?
this
And
yeah,
gives-
air traps inside in between the glass plate and in the gel.
Right.
So it should be taken care. So for that, edit imaging is very important. Then visualization of image, then construct match his hierarchically. Okay, this there is a procedure how to how to create a match set and how to create a class set. There are two differences. This we will discuss more in our software demonstration. Then processing gel images, where we can detect the spots and whether we can edit those spots, like how these are all the original spots or these are all the some dust particles, or else if there are couple of spots which is coming and showing as a single spot, in that case we can edit that particular spot, we can split them into two different spots. And if somewhere the boundaries, software itself gives very nice boundaries, if you are not at all satisfied with those boundaries, you can able to easily draw yourself whatever the boundary you would like to draw. This is what we can do. Then data analysis, once you did all these things, you will match your gels, and after that you can get some statistical data like ANOVA, and with the help of these you can see which are all the upregulated and which are all the downregulated and which are all the similarly present, and these are all you can see actually in this output.
So I guess preparing images for analysis, that is very crucial, because although there are a lot of claims from all the software that it's very automated procedure, but regardless of that, like I guess all the software pick up a lot of artifacts, even small dust particle and even stains will be directed as a sort of a small spots on the gel, right?
Yes.
So one need to have both automated as well as manual interference and a balance of each to ensure that whatever spots are picked up, those are the real spot. And I guess that's where what you mentioned, that different type of spot boundaries and the tools which can enable us to remove the spot or add a spot or drawing the right boundary.
Even I'll go through each and individual parameter also.
Those will be very critical.
Yes.
So I think it will be good to discuss those in more detail.
Yeah. In the image analysis workflow, first we'll go through the add images. How to add, one can add images to the software. There is a option when you open image pool, as you can see here. This is from there you can add any image which is stored in any portion, any area of your computer. Like once you add them, you can see it goes to that particular area, and the software can able to analyze .gel, .mel, .tiff kind of images. It very easily it can be analyzed.
So various type of extension files can be used.
Yeah, exactly. Various kind of types of, except the simple JPG or uncompressed compressed TIFF. These kind of gels we cannot able to edit. But other than that, .tiff, .gel, and .mel,
it requires high resolution.
High resolution images, so that it can have much information with this image.
A minimum 16 bit.
Exactly.
Okay.
At least the image should contain 16-bit resolution.
Right.
This is the main important thing in this software. During adding your images, it will ask you what stain you did for your gel. So we need to mention our color, especially they are within the software we incorporated different stainings. So those are like chromacy, silver, or different fluorescent colors which you have to mention. Once you mention that, then you can open your images. Now
just let me ask you one question here. So what is the use of defining the different type of stains? Is that going to change certain parameters based on the what type of stain are we going to define?
Yeah, by giving this particular staining, one can give a pseudo color for these images. So it will help you to viewing the images. This
So
is
if
what it.
you have a
it
stain
will
with the
help you.
Kumasi, you can see in the
Blue background
and with the silver and the brown and
brown
different
and
type
as well
of
as
stains.
as well as different fluorescent colors, different fluorescents like RBG. You
Right.
can see.
But it's just a pseudo, right? Like it has
Yeah.
stored a TIFF image in the grayscale.
Yes.
And that's what it's going to analyze.
Yeah,
But
exactly.
rest of the things are just for visualization and getting a feel for the type of stain.
Yes, whatever you are doing the editing of the visualization, that would be only for your visualization only. There is no changes will be happening in our data. This is the major important
I think
thing
that's
in
very
here.
important for people to learn, that even if they're playing with the image, that is not going to change the raw data. That
Raw
is
data.
important.
That is very important.
Because
yeah.
all it's just helping you to visualize the image and align it properly and
yes,
helping you in the analysis process.
Exactly, this is what during your analysis workflow there are few recommendations which we can give. Those are like use viewing tools like contrast, overview, profile, 3D image, as well as to get an idea about image quality. If once you can go through all these parameters, definitely you can decide this image can be analyzed or this cannot be analyzed. So once you get an idea of this, so you can go ahead in your analysis of
So it
your
means
data.
the user has to do this thing for each spot for say, right? For example, if there's any doubt whether this is a split spot or a real spot,
Yes.
one need to look at the three-dimensional profile of it,
Yes.
and also look at the contrast and just try to change different type of parameters
Yes.
to ensure whether one is really handling a small spot
Yeah.
or an artifact.
Yes, exactly. Even as we know, if any dust particle give you a spike kind of a three-dimensional view,
Right.
and one direct protein can give you the direct the perfect peak sort of thing. So this we can able to differentiate and we can
So one
decide.
can take a decision based on the
Yes.
three-dimensional views of these.
Exactly,
spots.
exactly. This is what we can do. How to adjust the contrast in our workflow. So you can see there is an option from view. There is an option to change the complete contrast and brightness, whatever you want. In that case, you can select any one of the box from the select tool. Okay, from after the selecting that particular box tool, you can change whatever you want from the x-axis, where here you can see from y-axis there you can see that is the contrast and as well as brightness. Okay, so you can see in the real changes you can see in this particular block. After changing the whatever the level of the changes you want for visualization, once you think this is perfect view, then you can press on apply. There is an option like apply. You can just click on the apply button, then it will the same parameter will be given for all gels. This is what it can be
So
done.
if you have a match set of, let's say, 20 gels,
Yeah,
so if you adjust it for one, it can be applied
for all
20.
gels. The same parameter can be applied for the all 20 gels.
Right.
Exactly. And after doing after setting this your contrast adjustment everything, you will see the final image like this. Still there are, you can see the saturation, you can see like in red color. So this can be removed, like again readjusting the particular
So these
contrast.
red dots which are indicating for saturation,
Saturation.
are actually generated by the software.
Yeah, exactly.
Software is indicating that this contrast parameter is not
not suitable,
perfect.
not perfect. You may need to change little more.
Okay.
But anyway, it's again for visualization.
Right.
It's not at all like it is not
Not
going
going to affect the image process.
image processing. Okay.
Okay.
A particular, you can, there is a zoom option in this. If you can see that particular area, then you can directly see that particular area in a big screen, so that you can see if is there any low abundant proportion, or if there are multiple spots binding together and showing as a single spot in 3D view, as well as you can see like this also. You can decide actually here, especially when manually one can draw the outline when we are zooming that particular image. So this is very helpful tool again.
Yeah, I guess many times when you have many gels and you just want to look at one particular area, it's not
Yes.
really possible to look at in a very small image. So you
Exactly,
need to have
exactly,
zoom tools for
exactly.
expanding the image.
Yeah, there is another tool in the software like profile view. there you can select that particular profile view. Wherever you goes into a particular spot, it give you that particular profile of in the x-axis as well as y-axis. You can see that particular. If I can select it, it can be we can be discussed more about during software discussion in live demo. Okay.
So on the x and y axis showing a peak, right? So
yes.
that peak is actually
which is a spot, which
so
is
for each spot when you click on it.
Yeah,
it
exactly.
shows the profile.
Yeah, no need to even click. Also, you just the mouse can go to if you move a mouse cursor to any other spot. It just wherever mouse cursor goes there, it shows that particular peak.
Okay.
Exactly. Then this is the another 3D view of a particular area. Here earlier which we are discussing a particular spot we can see as a 3D image, but if you want to see a particular area also you can see as a 3D view. This is what which you can see. Even with this also you can able to easily identify which are all the dust particle, which are all the real protein spots. This is what it will helps you. Now let us discuss about processing the images.
Right.
In here the first thing is we need to click on the spot detection. One should have to detect the spots. Even after the spot detection we need to match them.
So this process is automated, the spot detection process?
A spot detection process, anyway we'll go more detail in the next slide.
Okay.
But I'm just saying the three major steps in here. Okay. That is the spot detection, matching, quantitation. Okay. These three are the major steps. When we can see the spot detection, here there are three parameters majorly it will give you. Those are all like smoothness. In smoothness, it gives when you set up the lower smoothness, it oversplit the spots, like it try to split the spots as many as possible. When you try to give high value, it is undersplitting. So one can go through your image and how it is splitting. You have to set that particular smooth value.
It means if you are just trying to detect the spot,
yeah.
So first of all you need to give certain commands and parameters
exactly.
to the software itself.
Exactly. These are all the three parameters which
so
you can have
these
to
three
give.
parameters: smooth, saliency, and minimum area.
Yeah.
One need to define and in fact play with the
exactly
numbers.
Play with the numbers. That is there are optimum numbers. Like if you can say smoothness, 2 to 3 is quite enough. Okay. This is there is no much variation in this. But whereas saliency, this is very critical parameter where it takes the 3D image view and it sees the exactly how slope is there for that particular protein. If you are giving the higher slope, then it will give it takes the very probability. Okay. Then when you are giving the lower value, it will take the lower probability. Okay.
So higher the value, higher the probability.
Yeah, lower the value, lower the probability. In that way, it will take. Okay.
What about minimum area?
Minimum area. Actually, the minimum area most of the time it can be removed. It can easily remove the dust particles because any protein having the minimum area above 5 at least. So whereas we are putting 5 as a minimum area most of the times, maybe 90% cases we can put 5 as a minimum area. So it removes the all dust particles. Only it take into consideration only proteins. That is what we put minimum area.
So these numbers will be, I guess, more clear when we will really look at a demo of the software.
Exactly.
When you can actually show that by changing the numbers, what is the real output?
Real output you can see easily. See, during our spot detection, how it looks like, whether it is over splitting or it's okay, this is optimally splitting, but everything you can see on the screen, and you can decide whether these parameters are okay or not. One can decide easily. This is
so these red boundaries are software
generated, and these are all the parameters which you can see which we are talking about: the smoothness, saliency, and the minimal area. These three are spot detection. Once you put this earlier, you can see these are very densely it is detected. That means they have given some wrong parameters. After that, what they did, they given some optimal parameters. Now you can see it's perfectly detected and perfectly boundaried. Okay, this is what
So
which you can see.
I think main thing is, first of all, one would like that software should detect as many spots as possible
As possible.
in very accurate way, and then whatever is still left over, because I can see in the slide in your first image in the left hand side on the top one, there is some spot which we are not able to detect by the software. So now those spots can again be detected by the manual way by adding the spot.
Yes.
So that is, I think, the advantage of having this tool.
Exactly. Even here there is another option. I think that they maybe remove that particular area. That is what it is not at all. It's not at all detecting this particular right-handed portion area, because there is another option, exclude area. So one can exclude that particular area in analysis. So
it means if you are interested in only certain region of the gel,
certain region of the gel,
then one can easily
include that particular portion only.
Okay.
In this experiment they may be interested in this particular portion only. That is what they included this much data. This is what happens in this. This is the 3D view. Very clearly you can see this is the background automatically removed here actually, where because whatever the boundary which we are putting, when you can see the 3D image, it's not taking the bottom of the 3D, that is the protein peak. It is taking some portion, it is leaving some portion, and it is creating the particular boundary, so that the volume, total volume, it is going to take above the boundary only. It's not at all taking the below the boundary, so that we are directly eliminating the whole background. This is what it will help you.
So I think in the slide of the quantification of 2D, the way it is shown that the green circle which is defining the spot boundary, which is just a little bit above the background.
Exactly.
And so maybe it is covering almost 75% of the maximum peak of this whole spot.
Yes, 75% of the peak maximum.
Because you will expect at least 15-20% will be the background in the bottom part. So then whatever you
even we can put this particular 75% for all spots, so nowhere it is going to affect in your analysis.
So then assuming that 20-25% is the common for the background
for all the spots.
Right.
That is what it will be helpful. In analysis workflow, now we can see the quantification of the 2D gel electrophoresis. See, there are basically 4 parameters which we are seeing here. This background is automatically corrected, lowest value in neighborhood, then boundary, the defined at 75% of the peak maximum, so one can remove easily the background. Then volume is calculated above boundary level only, where you can see the green line, green boundary, this above of this only we are calculating as a volume. Then normalization through relative volume, that means this relative volume is the completely as all spots from all spots it is reducing this 25%, so remaining whatever the value is relative volume, this is going to be we are going to be analyzed actually. Compared in the analysis workflow, quantification of the 2D electrophoresis gel is one should look about the background of the gel as well as boundary of the gel, volume and normalization.
These are various parameters look into the
exactly. Then background is automatically corrected lowest volume in the nearhood. That means actually when we are boundary defined at 75% of the peak maximum, then volume is calculated above boundary level, so that you can remove background very easily. Then normalization through relative volume, you can easily normalize your spots.
So in this slide what you have shown, the green circle which is defining the spot boundary, that is actually shown from the 75% of peak maximum.
Yes.
So it means you are eliminating the background from 25%.
Correct.
But ideally that should be kept consistent for the whole spots. Right?
Exactly. Yes. This is the normalization thing which we are doing.
Right. So that there is no artifact in the analysis.
Yeah. Exactly. Correct.
And then one can remove the background artifacts and then only look at the peak volume and the 75% of the peak area.
Correct. Most of the time, if it is possible, avoid spot editing majorly. But you can do the spot editing, but when you are doing the number of gels more and more, in that case, if manually you are editing, in that time we don't know what are the whether the boundaries or whether the splitting. This you cannot do for all gels equally. So try to avoid this thing initially, but if it necessary, definitely you can do this. Then introduce because introduce a lot of subjectivity. Okay, this is there, and not supported with dye gels. It is not at all supported with dye gels. Then use alternatives that like composite spots by defining multiple matches and propagate spots from one match set to another. Like once you are matching, if those are all not matching perfectly, and the same gel you can try to match another time, third time, fourth time, fifth time, like try to match as many as possible, and you can introduce lots of like 4 or 5 landmarks, so it can easily matched as well as it can easily detected.
Okay. So your recommendation is that one need to rely more on the automated analysis.
Exactly, yes.
And because the number of gels, if there are more, it's very tough to do all the quality control checks with the manual way.
Yeah, exactly.
By defining various type of landmarks and by repeating the process several times, one can actually increase the precision of analysis.
Yeah, exactly. This is what it's which we are recommending. Then if you would like to edit your spot, how to edit the spot first? Then first of all, this is the way which we can split out the particular. There is a tool for spot splitting. Then you just draw wherever you want to split, then it can be created as a 2 spot now. Then in this way you can easily edit your spot, and as well as same if you want to merge 2 spots, then by this way you can draw your line from both sides so that it can be merged together.
So these are manual tools which can be used for splitting or merging the spots.
Yeah, exactly. Then another option in the spot editing, if you are not satisfied with your boundary, you want to enlarge this particular boundary, then no need to delete and redraw your whole thing. Instead of doing that, you can just mention what is the area exactly you would like to extend to the spot, so that in this way you can expand your volume of your spot. In the same way, if you want to decrease some portion from your spot, like whereas you can see there is a tail sort of thing, but you don't think so this may not your protein portion, so one can easily cut that portion, so they can reduce the spot size also. This is the thing
so
which
defining
you can do.
the accurate spot boundary is very crucial. And different type of software views like 3D views and profiles can actually help one to take that decision of what is the best boundary one can define for the spot.
Exactly. Then matching. Once you finished off whole your editing process, now this is the time to match whole gels. So we can place one or several landmarks to support automatic matching. So once you are matching, okay, it can be matched, but if you put then landmark sort of thing, it can be very useful to matching. I will show you images how it affects in matching. Then keep landmarks at minimum. Okay, the minimum, okay, if it is possible, try to minimize 2, 3 or 4, otherwise it will again create some discrepancy in your spots actually.
So I think it will be useful to have the landmarks in the various region of the gel.
Exactly.
So that one can actually align all the different gels by using these landmarks.
Yeah, exactly. Place on well-defined smaller spot which are clearly corresponding actually. So you can see a small spot, so that it may not if it is a little bigger spot, it may not take that particular area into this another gel also. Okay. So in that way, if as minimum as possible, try to put in a small spot. This is another important thing in this. See, once if you match this kind of vectors, you can see actually these are all the vectors which you can see exactly. What does it mean exactly? This is deviating from the reference gel to that in remaining gels, how it deviating that particular protein, whether it is one side deviating or it is differently deviating, that you can see exactly here.
So the overall trend of
the matching, it will show you. So there is no landmark at all. you can see once we put the landmark, how drastically things changed. There is no there is no tail at all.
Right.
Okay, that is what it will help you in landmarks. So this is another thing matched with one landmark. Okay.
So again, the vectors have now look well organized and
Yes.
the similar orientation
Yeah, exactly.
as compared to ones we have seen earlier where there was no landmark.
No landmark. After matching everything, so how to design your experiment here, everything, how to match the gel, how to create the match set, and all this we will see more in our analysis. Okay, our software analysis. This is what which looks like from image pool we have added our images, and after that we have to create our match set, and we have to create our class. This will more detailedly will go in during our software demonstration. And these are all the analysis workflow, the complete data analysis. After data, that is the once you get the complete data from that, what are the flow fold regulations as well as what is the statistical parameters, then you can compare all the parameters and you can select your interest of protein. This is what which we can do finally. Image master platinum software demonstration. Shall we start?
Sure. So I guess you gave an overview of the whole data analysis which one can perform by using the software, and different type of challenges one may face during the analysis, as well as what are the different type of parameters one can actually use for proper image processing. But I think it will be really useful to see the software layout and how one can really take these images for analysis. So yeah, please go ahead and show the demo on the Image Master Platinum software.
Yeah, this is the software layout where you can see where in your PPTs. This is the original software layout. Now here there is an option like image loader. Like add it. Wherever your gels stored, you can go to that place and add the gels. Like as I have multiple gels here, I'm going to add few gels to our software to analyze. Like gel 1, gel 2, gel 3, and gel 1, gel 2, gel 3.
So you are processing 6 gels.
6 gels at a time. These are all the 6 gels. Basically, 2 replications, like 3 replications for each gel actually.
Right.
That means one is control and another is the treated in that way. Now, as we discussed in our PPTs, this is the thing which is asking for particular staining. Here we can give chromacy or silver or different fluorescent. So as this stained with silver, I am giving this silver and I am applying the same color for remaining the gels. Now click on OK. Now as you can see the 6 gels at a time in this overview. After that, now you can see the proper spots in all images, very bright images of which you can see. So now we need to edit the images for visualization.
So we need to change the contrast
and brightness, different features. So this is the button where you can adjust contrast and brightness. So as I can show here, if you now you can't see any differences now after changing also, but one can have to press on apply. Another feature is available here.
So now more spots are visible and
more spots are,
sharp.
yes, more sharp. Otherwise there is an option that you can select a particular area, okay, then change the parameters. Now you can see real-time changes in that particular spots.
But is that going to apply only on one gel, right?
No, it's not. Where we selected, that is, you can see now, after you are satisfied this particular parameters, then you can apply. So it can be applied for all gels.
Okay.
This is what it will help you. So now we can see more spots very clearly here.
Throughout the gels.
Throughout the gels, everywhere. So this is another option. Now your spot, this visualization is clear. After that, now we should not disturb anything in our original images. So we need to make a copy of this particular images. So for that we can crop and create a new image, because in our raw images, as we have some artifacts in our gels, we can remove all those artifacts. Okay, like
So there are some gel migration issues,
gel
and if
migration
gels have not
issues.
run
Yes,
uniformly,
yes,
you can crop the images,
Yeah,
and then you
exactly.
can store a new copy of the fresh image without changing your
original image. Otherwise, once during your analysis you have changed up all the things in your original image, and you may losing your raw data.
Right.
This should not happen. So what we are going to do, we are going to crop them. So when you are selecting in a particular gel, it is going to select in a single gel. Instead of doing that single selection, just press on shift and control and select a area which can be common in all gels, so that at a time you can select in all gels.
You can select all the regions
in different gels. Okay, and this is what you have to do. Then you can adjust them the same way, so that after pressing Ctrl and Shift in the keyboard, you can easily edit all boundaries at a time. Otherwise, if you are editing a single boundary at a time, we don't know, we may doing some wrong in image to image.
Right, so manual judgment is not required because we can do this automated way. So all the processing will be uniform.
This is what we prefer. Then you can crop them. This is the crop option. You can crop them. Then do you want to create new images? Yes, we would like to create new images. It's a very quick process. So now you can see all edited images here with extension of bracket 1. Okay.
So same original image is stored and also created the new image
with the cropped. Okay. After that, you just close them. Then open whatever the edited images.
So now we don't need to select all the 12, but we can just
processed images. One can create a grid line on the gel. Okay, from the view option, they can create the grid line so that one can easily judge whether all gels are showing the reproducibility or not. In which grid I have the what number of spots I have, and remaining.
Also, it will help to decide which landmarks one need to use.
Yes.
As well as which areas one need to really tweak around different parameters.
Exactly. This is very helpful tool again. And these are all the another option is if you want to remove, and you can click the same way. And another which we can talk is show profile. Once you click the profile, wherever it goes your mouse cursor, it shows that particular spot.
Right.
Profile, how it is looking.
Both X and Y axis.
Yeah, X axis, Y axis. You can see for all gels the same thing here. So this is just initially we can see the gel quality exactly. This is what it is showing. Then you can remove that particular tool. Okay, this much is okay initially. Then now we are going to design our experiment. How exactly we have our design of our experiment and how we can manage our gels. Like from the project, we have to create a new project.
Yeah.
Okay, this is the project which we are doing is IIT demonstration.
Sure.
If you have any description about your own experiment, maybe you may have some something about specific details for that particular project. You can enter here, you can type here, and it can be stored within this project. Then click OK. The IIT demo already exists. Okay, so I can give IIT demo 1. So now in this IIT demo, one can create the match set here by clicking right. So match set is there are two gels, 81 and 82. This is the 81 is the control and 82 is the treated. So
you have control and it's 3 replicates, and
treatment it's 3 replicates. I'm giving 81. If you have again same description you can give for 81. Okay, and then create another match set, give 82. Okay, now you shift the gels where are 81 gels to 81 folder and 82 gels into 82 folder. like you need not to do anything, just drag them. It's a very user friendly. Just select
so those 3 images from one replicate you are moving in one folder,
and
other 3 replicates from other treatment you are moving in other folder.
Exactly. Yes, this is the, whereas you can see one gel has a red half and blue green half, and the same here in the 82 also the same way you can see. That means this gel set as a reference gel, and remaining 2 gels are going to be matched against this reference gel.
So can any gel be made as a reference gel?
Yes, exactly. Any gel can made as a reference gel. There is an option from the view. You can change the option. Okay. This is
so
what.
It means there is no bias here.
No
like
bias
one can
at all.
actually change the reference.
Okay. This is what. Now, display them. Now you have all of your gels here. Okay. Once you have the gels, we can directly detect the spots now. Okay. From this button you can detect them. Now the 3 parameters: smoothness, saliency, and minimum area. The smoothness, as we can see, if you are increasing the smoothness, it is going to under splitting, and if you are decreasing this smoothness, it is going to over split. So it is inversely proportional from the splitting. So it's a average value from 1 to 5 actually. So but one can set as a 2 to 3. It is sufficient. This value is sufficient for
to
start actually. And saliency, again this is also one can set up to 100, 150, but to start very good value is almost all 15 to 30. Okay. I am just giving 15 for this initially. Then minimum area 5 is quite good enough. If you think in your spot there are more spectacles, like there are much artifacts are there, you may increase this also. In that case, you can be remove very easily more artifacts. So in this case, otherwise 5 is okay, fine value.
So all the 3 parameters now one can actually accept those.
Exactly.
And then detect the spot.
Exactly. That is what the software detecting the spots. Now it's detected all spots in this gel. If you are satisfied with this particular detection, then it's fine. Otherwise,
maybe we should zoom in my region and see like.
Yes, exactly. I'm going to do the same. And now you can zoom this particular region. See how this spots are boundaried, as well as how the detection is there.
Can we apply the same zoom in parameter for all the 3 gels?
Yes, definitely. First come back to original. Now again
let's select one region and then apply that for all the gels.
No need, you just select like the same region. It is going to got it.
Right, I think it's very useful to see this way, because now
exactly
I think the parameters are very well defined.
Exactly,
because it has properly defined the boundaries. And there are some spots where one can actually now tweak around to get
exactly. If you want to edit these particular spots,
right.
Whereas you can see there I think you can't see there is a protein, but it is showing as a protein. One can delete this kind of spots also.
Right.
Like one should have to go to edit the spot. There is an option like edit enable.
Right.
Go there and just delete the spot. Wherever the spot you would like to delete, this particular spot I am not interested in this, then I can delete it.
But before we take the decision, can we just have a look on the 3D profile
yes,
of this
definitely,
one to ensure that we are not actually deleting a
yes, definitely
real spot,
definitely. And then reports, go to 3D view.
So the spot which we are interested in discussing, that is highlighted with the green boundary.
Green boundary, but it is only present in this particular spot, but it is not there. But we can see some portion here actually, that particular spot. See, but that is why it is detected there also.
Right. So I
Yeah.
think sometimes it is possible like your
yeah.
treatment may have that spot
Yeah, exactly.
appearing due
Yeah.
to the upregulation of a protein,
Yeah, exactly.
or it is totally shut down because
But
of
as
that.
but it is not the case exactly here, because these are all the 3 replication of the
Okay, right.
particular same protein.
So this should be the same.
This should be the same, but this somewhat it is not there, so we can delete these 2 spots here exactly.
Or in this case, I guess it is doubtful, right? So
Yeah, it is doubtful, or let it be. We can then finally we can see the statistical data whether it is there in both gels as well as treatment also,
Right.
so that one can go with the statistical parameters.
Right.
This will be helpful there. So no need of deleting also in
Now
this
let's
case.
look at in detail about one of the real spots.
Otherwise, I can visualize this thing. I can more zoom so that one can see more. Yes,
So now this is the intense spot.
intense spots which we are visualizing. As
Another boundaries are marked from the 75% from the top.
Yes.
Now let's say we can actually zoom out and
Yeah.
go back to the whole gel. So let's say if in a region we have missed out some spot, like software has missed out,
Yes.
then there is an option to add the spot as well.
Exactly as well. This is the adding. Like let me zoom. Let me select a particular area. I'm interested in this particular area. I'm selected everywhere, and let me zoom that. Now you can see this particular area in all gels. Okay.
Right. I think it has defined the spot boundaries quite well overall.
But yes.
So that's why it's better to rely more on the automated.
Yes, exactly. That is why.
Automated detection.
Exactly. This is what. If you want to if you want to include some spot wherever, if you are interested, you can definitely add. Like this kind of extra portion, we can as easily we can delete actually. Like we can reduce this particular portion.
Second better ways to remove the spot and then draw the spot.
Yeah, that is also better way. But
Yes.
both ways you can do actually. Yes, you can delete it or you can redraw that. Is both as well as we can do. Now we find all spot boundaries are okay and everything is fine. Now we can match these 3 gels, how our reproducibility of our 3 gels, how reproducible of our 3 gels. Okay, this is what which we can do here. So come to your original state without zooming and match them. This is the match option.
Okay.
You can match them.
So now you are applying the same analysis parameter on both control and treatment gels.
Yes, exactly.
And you are matching the
Yes,
gels.
exactly. Now we are matching only control gels only.
Only one group.
Only one group. How our reproducibility is there within one group. This is what we have to check initially.
Okay.
So if you have, suppose maybe instead of 3 replications, if you have maybe 5 replications, maybe 6 replications, out of 5-6 replications, one can remove that particular gel and they can take remaining 5 gels.
So if there is some time during various experimental run, if there is 1 or 2 gels which are very bad, which are going to affect the overall reproducibility.
Exactly.
So by looking at this type of parameters, one can decide, okay, out of 6 gels, maybe 1 of that is very bad and going to affect our statistical parameter.
Exactly.
So maybe one should remove that.
Exactly.
And then select those ones which are having the very good number of matching.
Yes, that should be done. And now we can see in these 2 gels there are few vectors which we can see. This we can able to remove by adding the landmarks out of it. Okay, now this is the landmark option. Okay, you have to add your landmark initially in the reference gel only. Like if suppose you can think this is a landmark sort of thing, I'm just going to add a landmark here. So I have to adjust this landmark to the same position in remaining all gels.
Now vectors are, I think, improving
yes,
based on the landmark position.
Now again match them.
So from 146 it has increased to 746.
Yeah, exactly.
By increasing the
by adding
a landmark.
A landmark.
And now vectors are matching well.
Yes. See.
Right.
But you can see there are few
vectors
but still there. But these are all moving at the same side. So that means there is not like irregular.
Also,
So
I think
regular
one
way,
need to go through each
go through
region,
each
very
region of
carefully.
yeah, very carefully. So if you can add some more one more landmark here, it can be removed very easily.
Right.
This is what we can do. Okay, now we can save this particular thing and apply the same parameters to our treatment gel also.
I think that's important because
Yes.
you don't want to make any changes
Any
between your
change-
controls and
treatments. So you can see no need to remember also the same parameters. If you can detect your spots, it automatically gives the same parameter earlier which you all have which you have used.
Yeah.
Okay, the same smoothness is 3 and saliency 15, minimal area is 5. So no need to change anything. Then click on OK.
So is it a long process of analyzing it, or rather I think sort of quite quick?
It is very quite quick. Then it's completely finished. Now the same way you can you can go through each and individual gen by zooming and 3D view, and you can select which are all the spots and which are all the not spots. Then you can directly match them. Now I'm doing the same matching. 1274 matches have been done. So these gels are more reproducible. That means as compared to our
Other
control.
set.
Yeah. See, there is no even vectors also. Without giving landmark also, there is no vector.
Right.
So this is very fine gel. So now the what are the what is the replication which we did was confirmed. Okay, these replications are okay. Now one can analyze in the class analysis in between these two classes, like interclasses and intra-class sort of thing.
So between a control and treatment group,
Yeah.
one can actually now do the statistical analysis.
Statistical analysis. So now I am creating the classes here. Otherwise, before that I would like to go to one report point. What is the gel table? Okay, that is the these are all the gels which are present here. Shift A, you can select all the spots. Go to spot table. See now.
So what parameters are displayed here in the spot table?
This is the file name and the spot ID. It has given a particular spot ID for all proteins, and this is the pixels. Or it is the sorry. This is, let me. This is the match ID. That means out of 6 cells, this match ID which is giving. One second.
Let's continue quickly. Like what are the?
This is the file name. Thus, this is the gel number. Then spot ID, which is given complete number for all spots. Then match ID. Then this is the pixel size and x-axis and pixel at y-axis. Then if you can be able to give the PA also, and you can calibrate the PA as well as molecular weight also. Then intensity of particular spot and area of that particular spot and volume of that particular spot and percentage volume of that particular spot and saliency of that spot. These are all the things which you can
So all
see in
these
your
parameters for all the gels
all
can
these
be obtained from
the software. These are all the parameters which you can see in the software. Okay, so if you are satisfied with all these values, then you can go ahead your next level of analysis, that is class analysis. Okay, now I'm going to create a class here. Create a class. This is the 81. Another is the 82, which is control and treated. Now again you can shift these gels into 81 to 81. Another match set which I am creating, so where we can easily move easily before going to class, so that
So you are moving all the 6 gels.
All the 6 gels into composite match set. So all 6 gels are here, and now you can create again the same way classes.
So by adding different folders, I think one can actually avoid any sort of carry or mistakes in previous one.
Yes, exactly.
And
So that
all the steps are stored.
Yes, all the steps are stored. Now again, I'm shifting all 82 gels into 82 folder. This is the class folder again. 81 folder. Now
this is for the class analysis.
This is for class analysis. Now we can see both gels together. Now 3 gels from the control and remaining 3 gels from the treated. Okay.
Right.
This is what which you can see. Now it is behaving, it is taking values as a single gel of 81 and single gel of the 82. So now it is going to average of these 3 gels, and now it is another averaging these remaining 3 gels. So a single value you can get from the control, another single value you can get from the treated. Once you did the same thing, then quickly match them again. One can remove all these vectors by adding the again our particular landmark. So
same
this
process
is the same process what we did earlier. Now go to reports, where you can see the analysis classes. Go to table. Now you have the match ID. The match ID is the particular spot number as well as the maximum area. This is a complete maximum. Whatever the maximum area, as well as this is the 81 value and 82 value. Okay.
So for the control and treatment,
yeah,
both groups we had 6 gels, right?
Yes.
So what are the values in each of the 6 gels for each spot?
For each spot, exactly. This is the averaged value again.
Average of all the 3.
All the 3 gels
from each group.
Yeah. Ratio value, if you can see, this is the ratio value which is giving the upregulation or downregulation. Okay, and I think we need to add. Yeah. Now this is the statistical parameter on
so
the basis of
software gives you option for doing different type of statistical analysis.
Exactly. So this is the ANOVA value which we can see. On the basis of this ANOVA value, we can easily select our interest of protein, as well as you have the fold regulation also. This is 1 fold upregulated and this is the 1.2 fold downregulated. The same way you can see the whole values. Now no need to go each and individual spot. You can filter them easily. Now what we are going to do is, first at least ANOVA value which we are going to take. The maximum we are going to take is maybe 0.05 as the statistical significant value. So those many spots it can be highlighted. Okay,
right.
So this is the spot which is already undergoing this particular
ANOVA value
threshold, and it can see all the spots which you want, which you have this particular ANOVA value. Those are all highlighting here. Right. Now it can be very easy, one can easily go through that, and this particular table you can easily export to Excel also. From there you can have your complete data also. This is what the complete analysis software gives you the output.
So I guess it was very useful to see like different steps what are required for performing this analysis. And also the software gives you a lot of options for doing different fold change and statistical analysis of how significant data is. And then one can actually still go back to those spots which software says is significant and look at manually each spot to verify that is a real spot.
Yeah.
So thank you very much, Srinivas, for very useful discussion and demonstration on the software for image processing on 2-dimensional gels. We'll continue our lecture flow for further 2-dimensional difference in the electrophoresis in the next class. So discussion with Dr. Srinivas on image processing by using Image Platinum software. I hope it was useful for you to understand the details required for image processing and how various type of parameters one need to pay attention during the analysis. So in today's lecture, in summary, we discussed about the challenges for the image processing which are associated with 2-dimensional electrophoresis, how one can process the images very efficiently, how one can analyze these images stepwise by using various type of software. Most of the software work with the similar principle, and to give you a feel for the data analysis, we showed you a software demonstration during the discussion with Dr. Srinivas from the G Healthcare software image processing by using Image Platinum IMP7. At the end, you must appreciate that one need to do different type of statistical analysis to ensure that the identified spots can be really further pursued for research, and those are going to be further processed by using mass spectrometry. And if they can provide some meaningful information, then one need to have full confidence. That is why we need to do statistical analysis beforehand. So, I would like to acknowledge Dr. Srinivas from G Healthcare for discussion on image processing and data analysis, as well as giving us a demonstration of the software for 2D gel analysis. In the next lecture, we will continue our theme on gel based proteomics, and I will introduce you with a new technology on 2-dimensional difference in gel electrophoresis or 2D dye, and then we will continue our lectures on 2D dye and its image analysis.